That tells me a lot of the nutrition. But you're not going to
Okay, so 11 o'clock, let's have a class. We will continue for the section 4, the slides. We hope to finish today. Next week we have 2 lectures. So we're going to do the review, and we'll have our first exam of AEM 341 on September 18th. So that will be roughly, we say 2 weeks from today. Is that correct? So just to remind you that. Again, let's do the review what we talked last section. So we talked about simple stain, gram stain. Then we said there is some special stain we need to cover. So the special stain what we covered is acid fast stain. And we talked about endospore stain. And today, every lab will practice the endospore stain. And we also will talk about endospore a little more detail. And we also mentioned about flagella stain. Today we'll talk about flagella. And then we also have a capsule stain. And then we will introduce capsule to you today. So that's we finish about the staining technology. Then we spend quite a time talking about electron microscope. And we said the electron microscope is a big difference compared to light microscope is the energy resource. The energy resource is coming from electron beams. And everything has to be in a vacuum system. And every material, what they have, the lens, is called electromagnets. The reason is that in the vacuum system, the electron cannot collide with air molecule. If it's collided with air molecule, then the image will be disqualified. We can say that. So we mentioned basically scanning electron microscope. We look at the surface, transmission electron microscope to see the internal structure, and we briefly mentioned about the specific staining technology. We call it a coating. It needs to be cut into very thin area. We call it shadowing, and we're using a heavy metal like a lead to finalize it. So this is we've said briefly. Then we have 2 slides talk about the morphology and also the arrangement of the bacterial cell. And you already see these during your practice of the gram stain. So what type of the arrangement, what type of the morphology it is. So this is what we pretty much finish the last slide section. Then we move on to the structure of the bacterial cell. And the bacterial cell structure, we first of all said the big difference comparing between prokaryotes and eukaryotes is cell membrane bundle system. And then we spending quite a time talk about the nutrients transport. The nutrients transport mechanism for the bacteria have like passive diffusion. We talked about facilitate diffusion. And we talked about active transport, or we say active diffusion. I said these for bacteria does not use too much. For bacteria, they use quite a lot, is group translocation. And it's rely on PEP. phosphoenolpyruvate transferase. The reason is all these chemicals, especially the carbohydrates, when they come into the bacterial cell, it's already being phosphorated, which means they carry a phosphide and then a phosphor, and then they can directly go into glycolysis, or we say modified glycolysis. So I say this is very important. And then we talk about these chemicals a little bit. Later on in exam 3, we're going to talk detail about these chemicals. So that's what we finished last section. So today we're going to continue. First of all, it's kind of like a review. Bacteria cell wall. So we already practiced the gram stand. We already talked about the gram stand. Obviously, because of the bacterial cell wall, we could differentiate them into gram positive and gram negative. So gram positive is purple, gram negative is pink or red because of sick peptidoglycan. That's one of the reasons we call it a cell wall theory. Now, cell wall function, you can list all of that. Let's say protect osmotic lysis, give the shape of the bacteria, protection toxic materials coming inside and contribute to the pathogenicity. All these things is cell wall functions. Well, we're going to spend in time talk about this guy. So we already said the major difference between gram positive and gram negative is cell wall structure. And the cell wall structure, lots of that are the same. The big difference is gram-positive bacteria have a very heavy peptidoglycan. And the gram-negative bacteria is very thin. So we say gram-positive bacteria is thick, and the gram-negative bacteria, it is thin. So what is peptidoglycan? You can look at the slides. It sounds like kind of like the complicated, or you say messed it up. But if we want to say very simple, what is peptidoglycan? Peptidoglycan, it is composed by modified sugar plus linked with amino acids. If you said it very simple. So two chemicals major is modified sugar. A sugar also we call carbohydrates because CHON, is that right? Amino acids. So two chemicals, sugar and amino acids. So the question will be, what are the modified sugar? And how they connected with each other? The modified sugar, basically, N-acetylmuramic acid and acetylglucosiamine. So then we're going to show you how they grabbed together, how they grouped together. So you have a glucose. And this is everybody pretty much know. Okay, I may have got a jar right here. So glucose, H, OH. This is still H. If it's OH, then you go OH, H, OH, H, H, CH2, OH. So I hope all you guys remember the structure. No matter which classes, from biology, biochemistry, organic chemistry, you're going to have to know the glucose. This is what the glucose looks like. Okay, what means N-acetylglucosiamine? Which means something will happen right here. They connect to this guy. That's N-acetylglucosiamine. Now, what is N-acetylmuramic acid? Let me go it again. CH2OH. That happens right here. This guy comes down. This is go. Go here. Then what you do? CH3 H. That's muramic acids. Now lots of the time, this is also with this again. Muramic acid. Now why these are important? Because of this. What we call it in the chemistry. Carboxy group, is that right? They could connect it with amino acid. The amine group. Now we're going to go back to see what is the amino acid looks like. What is the amino acid looks like? So amino group, carbo C, H, carboxy group, and the R. So depends on R. You can have H is glycine. Then you can have CH3. That's alanine. What else you have? You have like a maybe like a CH, like a SCH2, CH2. That is called methionine. We're going to talk about later. And then you may have something like this guy. Okay, CH. So it's phenylalanine. Phenylalanine. We're going to talk about this in the lab. So that's amino acids. So these things they can connect it together. How they do? So let's say how they connect with each other. So let's draw this one right here. Okay. We draw one here. We're going to draw another one here. Briefly, okay, not going to be too complicated. Let's say this is N-acetylglucosamine. That means here you're going to connect it with this. How they connected? This is N-acetylmuramic acid. How they connected with each other? Biochemistry you learn, chemistry you learn. With glycosidic bond, they are called 1,4 glycosidic bond. They connected with each other. Okay, how they connected vertically? Then remember here, this O going to go here. Then you have CH3, you have H, you have C, OH. And we said this guy could connect it with here. It's a peptide bond, looks like similar. So what we do? When you go down here, you become NH. Then you go here, you have H. Then you have H. You have C comes here. Lots of things. And then here you could grab another one. Is that right? You grab another one, you can draw like this. Then you grab another one, so you can draw like that. Become long and long. So NAM and NAG. So become longer and longer. They also could connect with this. In a diagonal direction. So what are they? If you really want to draw, that means it's NAM in a alternate sequencing. And then they could connect it with the first one. People already know it's alanine. And second one is called D-di aminoylic acids. Mylic acid. And then the next one, it's glutamine. And the next one is still alanine. Okay, then you have NAM, then you have NAG, then you also have NAM, you have NAG. The same thing, they connected with each other, the same thing, with alanine, with glutamine, with D-diamino, with alanine again. Now, how they connected with these? This is connected with glycine. That's how they cross-link. Now, why we want to draw here? The reason is, which gives you an impression, peptidoglycan, the bacteria needs the time to building. So they need the time to building during their growing status, and it needs lots of material. So the first time people find the most effective chemical to killing a bacteria is going to have to work on these chemicals. Okay, you can block here. You can cut here. If it doesn't work, then you cut here. If it still doesn't work, what it means doesn't work, which means antibiotic resistant. Then you can cut here. If you say all these things are really bad, I will not let the bacteria transport the energy resource, which means the material to building these chemicals. That's another way to prevent the bacterial growth, to inhibit the bacterial growth. So the draw here, the idea is to let you know the idea how they connected with each other. It's very similar to the amino acids connected with the peptides. But you see with sugar, with amino acids, they need a time to build it. And at the time, it's the best time to using antibiotics. So you will be finding most of the antibiotics, when they kill bacteria, is better for the bacteria is growing. Especially for the bacteria is using material to building peptidoglycan. It's the best time to killing bacteria. And you also will find most of the commercial antibiotics are more effective for gram-positive bacteria than gram-negative bacteria, because gram-positive bacteria have this sick peptidoglycan, and the target is more obvious for the drug to attack him, and the gram-negative bacteria is very thin. So imagine if all the antibiotics are really very effective for gram-negative bacteria, then E. coli 0157H7 will not be on the headline of the news media. You heard about it all the time. There is a 500 pounds or 5,000 pounds of the beef are recalled in like a plants in Indiana because of the E. coli 0157H7 contamination. For example, there is about 5,000 pounds of the chicken thighs are recalled because of salmonella contamination. That's the reason. Because those gram-negative bacteria, it's very thin, so it's hard to do the damage. Okay, so that's why I want to draw here, and this looks very simple. It just gave you the idea how they connected with each other. This I already draw, but give you a very good picture from the McGraw-Hill textbook, which is showing you the plasma membrane system, the fluid mosaic model. You have a periplasmic space, and that space, it is also important because it's get ready for the transportation of some of the enzymes to be activated in the cell building system. Then you have a heavy peptidoglycan. You can see it's cross-linked with modified sugar and amino acids. And on the top, you have some of the other chemicals, like teichoic acids, like lipoteichoic acids, but the function is not very clear. It's still unknown. And because of the lipoteichoic acids, So gram-positive bacteria, more or less, still have some lipids, like 1 or 5%. That's what we talk about. Okay, gram-negative bacteria, you see the difference. First of all, double layer. You have inner membrane system, outer membrane system. Peptidoglycan is very thin. But the tricky thing is that they have something extruded out called LPS, lipopolysaccharides. That's really bad, and we talked about already. Lipopolysaccharides. If you see more clear here, I have lipids A and the polysaccharides. So I will mention again later on. You heard about this in the news sometimes. What this means? What is O mean? That is O antigen. That is a polysaccharides of LPS, of lipopolysaccharides. That's why O you can do it. Now lipopolysaccharides, I already said, it will have endotoxin. Now, good thing for the endotoxin, it is heat liable. What means heat liable? Which means you cooking, you can kill it. So that's why when you buy a ground beef, you look at the label of the package. It always have a thermocouple there. Said you have to be roughly cooking to the internal temperature like 160 degrees Fahrenheit. The reason why they can label that, because roughly cooking, you can kill the pathogen. You also can kill or destroy the toxin. But the other way, there is another toxin which is called exotoxin. That's another story. Exotoxin, like Staphylococcus aureus, will create it. That is the one is not heat liable, which means even you cook them, the bacteria is dead, the toxin still could be there. So we will mention that. That's also the difference between food infection and food intoxication. Okay? So that's something I want to say. You can see here very clearly the difference between gram-positive and the gram-negative bacteria. You can see the lipopolysaccharides, the lipids A and the O polysaccharides. Okay, so this is a cell wall structure will be mentioned. It is important, but the key thing is you know the peptidoglycan's basic structure, what it looks like, what's the difference between gram-positive and the gram-negative cell. These two slides are also important because I will have a question in the exam for you to label those structures. But we will have a practice question for you to do before we do the review section. But I just want to let you know, because this is important. This is a key thing for the first section of the lab and also the lecture. Capsule. Okay. We already talked about and I said Streptococcus pneumoniae. You see this white area is capsule. It is a polysaccharide. It looks like, what it looks like on the other plates. Look like the cheese smear. Especially here. The given example is KPEUMONIAE. That's Klebsiella pneumonia. Klebsiella pneumonia, it's not a major pathogen for pneumonia, although it says that. But what is special? It had a very heavy capsule. So Klebsiella pneumonia, when they grow on the agar plates, it looks like the cheese. The colony looks like cheese, very sticky, because the capsule is accumulated. You can use light microscope to do, and we can also use capsule stain to do. But for the bacteria, the most important is, this is also a pathogenic structure. So what happens is, if a bacteria, there is some of the host cell there. Okay, if the bacteria come in, they think about the host think about some bacteria out there. Let's say a bacterial cell, and you have a host. And the host usually will be started their non-specific immune system, which means they're going to grab white blood cell. When they grab a white blood cell, they do the first thing is you're going to package it and generate a special structure called a phagosine. Phagosine. Once the phagosine formed, it will release lysozyme. And the lysozyme could cut the bacteria of special structure, which is attacking 1,4-glycosidic bonds between N-acetylmuramic acid and N-acetylglucosine. So they can kill bacteria. That's what happens when the host cell come in. When the bacterial cell go into the host cell, how the host cell do the defense, do the natural defense. Now the bacteria, they can survive or they can cause more problem because they have this polysaccharides, which is called a capsule there. Once you have a capsule there, this phagosome is not easy to forming. The releasing of the lysozyme cannot directly attacking here. So, capsule for bacteria like Streptococcus pneumoniae, those pathogens, it is a defense system. So we say resistant to phagocytosis. It is called anti-phagosine. That's what it looks like. The whole process here we call it phagocytosis. And when you have a heavy capsule, you can against it. Okay, so that's why the capsule for the bacteria, it is important. That's why Streptococcus pneumoniae will be first #1 community-acquired pneumonia pathogen in the United States. Now, in other words, since the capsule is that heavy, is that obvious? For bacteria, it's a defense system, but for us, we can use it. We can extract it, we can purify it to make vaccines. So some vaccines coming from the capsule. The vaccine against the Streptococcus pneumoniae, which is recommended for 60 years old or young kids to do the injection annually, which is coming from the capsule of Streptococcus pneumoniae. So capsule for bacteria is a defense system, is a protective structure. As a microbiologist, we could use it to make a vaccine. Okay, so there's a good thing for that, there's a bad thing for that. It depends who did it. Next one, ribosome. First of all, ribosome, both gram-positive, gram-negative bacteria has. Not only for that, bacteria and fungi, prokaryotes and eukaryotes, they all have a common machinery, is ribosome. What is a ribosome for? It's generate protein. So that's a place for protein translation. DNA translation, generate the protein. There are two major subunits. For bacteria, the large unit is 50S. The small unit is 30S. Add up together, it's 70S. Not 80S. Okay, why? Because S, the Vandenberg unit. What is the Vandenberg unit means? You have a cell culture, you have a protein, you do 5,000 G centrifuge, or sometimes more than that, maybe 10,000 G centrifuge, and then you're going to spin down, they're going to accumulate it here. The unit used to see the velocity of the spin down or precipitation, we call it the Vandenberg units. So, the whole structure of a ribosome, it's 70S. It's not simply you do the math. Okay, always remember, it's a very simple question in the exam, but some of you will still miss it. Now let's talk about the inside, 50S. You have another two things, which is 5S and 23S. 23S subunits have a very important structure. Is RNA transferase. It's an enzyme. 30S subunits have 16S rRNA. Is that right? That's important. 16S rRNA for some of the bacteria have V6 to V8 conservative sequencing. That's uniform for all the bacteria. That is why very popular. You heard about 16S RNA or RNA biotechnology to identify bacteria because of that. Some of you work part-time in the WVU Health Science Center. Then you do some of the research, extract the DNA, RNA, 16S RNA. What that comes from? It comes from here. Because there is a very conservative sequence, uniformed for all the bacteria. Okay? So you can see here very obviously, the picture here showing you the structure of the ribosome, what it looks like. And all these details we will mention again in our exam 3 when we talk about the DNA translation. Okay, nucleates. Nucleates, it's very long in the bacterial cell. It's usually in part of the chromosome. So the chromosome is very long. If you take out of the chromosome, it could be larger than the bacterial cell itself. Of course, it's a combination with the interaction between the chromosome and some associated proteins. Nucleates could be circular, could be linear. There are all kinds of things we can talk. But basically, very simple. You have a DNA and you have an RNA. That's what we're going to say in the exam 3. So, what's the basic structure? Very simple. You should know right now. 5 carbon sugar. We call it ribose. And the ribose, in their position 1, you are connected to a base. Is that right? The base could be A, T, G, C. It could be adenine, thymine, guanine, and cytosine. For DNA and for RNA, you have A, C, G, but instead of you have T, you have a U, which is the uracil. That's for RNA. That's a base. And then RNA and DNA, what's the major steps here? Is this 2' hydroxy bonds. If you have the O, we call it RNA. If it doesn't have the O, we call it DNA. That's why the D means deoxyribosome. And then you go here, you have this guy's 3' hydroxy bonds. It is important because it's going to connect it to have the nucleotides. Then more important here, you have CH2, you have this guy. This guy here, that is phosphate. So what is the DNA and RNA? It's a basic structure. It is phosphate with 5 carbon ribose with a base. So 5 carbon ribose, where they come from? If you know where chemistry comes from, pentose phosphate pathway. So for me, if you really know, it's a very boring chemical. Because you only have phosphate. You have a ribose and you have a base. And the structure already know for maybe more than 200 years. So DNA is a very boring chemical. Although we do lots of work. Compared to protein. Is that right? Protein, we have amino acids. We have peptides, we have a primary, secondary, tertiary, and then the cautery structure. We have a folding alpha helice, beta sheets, all kinds of things. So just want to give you some brief introduction. What is the DNA and RNA looks like? But detail we'll talk about it in the exam 3. But here is something very special for a bacterial cell. They have, you have a chromosome, for example. Okay, then the chromosome could containing something there. We call it an episome. A small sequencing is right here. We call it an episome. Episome containing some of the very special information. We also call it like a small linear DNA sequence. That is called a plasmid. Plasmid is very small, sometimes 20 to 100 BP base pair. It could be do replication, transcripting, translation independently, or it could be integrated into the chromosome. But it gives you lots of the trouble. If the plasmids can work it independently, then they carry on antibiotic information, then that's really bad. We call all plasmid. And all plasmids could make it universal antibiotic resistant, especially if a bacteria eat it. And we say transformation, which means a bacteria naturally taking a plasmid from another bacteria. Transformation is one of the way for the bacterial genetic information communication. We call it transformation, we call it transduction, and conjugation. Is that right? Those are the three methods. Now, transduction and conjugation will rely on bacterial phage. We're going to mention later. This rely on phylline or pili, which is a sex pili especially. So, plasmids is very unique. It's a closed circular DNA molecule. Sometimes it's linear. It could be work independently or integrated into the chromosome, but it's always carry on some special information. If it's carry antibiotic resistant information, then it's going to cause a problem. Related to that, we specifically, the research has been done, find all plasmid. Some of them has class 1 interrogate. This is widely existing in all the antibiotic-resistant bacteria. So back in 20 years later, when I was a PhD student at Colorado State University, we get a sample from Rocky Mountain National Park. All those national environment samples, the soil, the leafies, even animal poops. And you see a huge number of the class 1 interrogates. I believe here you have a lots of the state park at West Virginia. If you're interested, if you get the sample from there, you could also see lots of those antibiotic-resistant plasmids in their class 1 interrogants in the sample from the national park, from the state park even. Okay? So that's the plasmids what we talk about. External structure. We talk about the cell envelope. Then we have some other things we want to talk about is phylli, fimbrial, and flagella. This is the phylli. Sex phylli we already mentioned about. Short, thin, hair-like. It's appendages. Some people say it has some motility capability, but I will always say this is small. It's not their major function. The motility, the major function is flagella. it is not for motility. DNA uptake, yes, because it's for conjugation. But more important is transmission of the genetic information. So finally, the most important thing is related to the sex phylum, which is transferring the genetic information. Part of that will be assisting the plasmids to get the genetic information. So they have a combined function of transformation and conjugation for lots of the bacteria. Flagella. This we already talked. Just to be a little brief here. So flagella, composed by flagellum. It is a structure for bacteria doing the motility. We call true motility. Now, based on the structure, the location, and the number of the flagella, there is 4 different categories. If you only have one tail or one flagella on one side of the bacteria, we call monotrichous. Which bacteria is typical monotrichous? Pseudomonas. Pseudomonas is a sporage bacteria, and I said earlier, like Pseudomonas fluorescens, you bought a beef or especially fish, you unpackage it, you put in the refrigerator about 7 days. You see also small dots comes out, it smells really bad, and ammonia comes out. The bacterial sporage is dominant with Pseudomonas, but it doesn't have a very strong motility function because the monotrichous. It could be like this. Have the flagella on 2 ends. That's called amphitrichous. The bacteria like Listeria monocytogenes. Some genes looks like that. E. coli, part of like that. A little bit strong motility. It could be stack on one end, Liphotrichous. Which bacteria? Lactobacillus. Using for fermentation, has kind of better motility. Sphaerium, which is in your slides, could be have a strong, relatively strong motility. But you compare to this guy, it is really a small baby. This guy, if you see a bacteria, the flagella looks like that, the last one. This is Proteus vulgarius. Then it has a very strong motility. Because this one, we call it a petrotrichous. Which means, what means petro means? That means it's almost covered the whole surface. Looks like they have wings everywhere. Then they can go everywhere and have a too strong motility. We can use a very simple media to test bacterial motility, which is sulfide endomotility agar. Basically, is the agar made by 0.5% agar. And then you put the bacteria right in the middle. And then you do the incubation. If after 24 hours, you see the bacteria looks like that, that's possibly petrotrichous. If you see the bacteria just stay in the middle, that could be monotrichous. So it's very interesting when you have these flagella. We can see some of them has a good or bad motility function. Okay. Relatively quick, we're going to talk about Treponema pallidum, which is spiral sheets. We already said it's a very special bacteria. It's asexual filaments. The spiral sheets usually coming from the cuts, butts, and abrasions. We need to use dark field microscope to do the observation. If you still remember, then what it looks like that. And this is a very special movement structure. It's also called a flagella, but not true flagella, called endoflagella. Corkscrew-like movements, very special for spiral sheets. Typically Treponema pallidum. Okay. Now, the bacteria has a spiral sheets, has a flagella. They can do the movement. Now, why it is important? Because they could do something very special called chemotaxis. When you have in the chemicals, if the bacteria wanted or say attracted, they will go. If you have something which they think it's harmful for them or like harsh environments, they will flee away. And the people has been done using the capillary tubes to see the difference. But here is the kissing. What is the kissing for that? The chemotaxis is a round trip. You go and you come back. Like you go to the airport. You're not going to do one way. That's lots of the study has been showing the chemotaxis is a round trip. Okay? So that's really why flagella is important. Endospore. We want to spend some time talking about endospore because we have 3 labs to do real quick, and we can skip some time in the lab, talk about those basic knowledge. We can talk about the endospore and give you some of the background information. Bacteria has endospore, but does not mean every bacteria has endospore. Let's say E. coli. E. coli doesn't have endospore. Salmonella does not have endospore. We call it a non-endospore forming bacteria. But some bacteria special has endospore. Basically, is Bacillus and Clostridium. Bacillus have 2 species, subtilis or cellulus. Clostridium tetranym, Clostridium difficile, Clostridium perfringens, and Clostridium botulinum, which is called botulism. You heard about that. These we all going to cover in the exam too. Maybe it's 3 weeks from today. This is biosafety level 3 passage. Only limited facility in the United States we can do. This pretty much common, biosafety level 1. Bacillus is also very special. It's a thermo-resistant bacteria. It is also has a special enzyme called use starch. So we will do some work in the lab. It is also a very good quality control pathogen, quality control bacterial cell for like autoclave, do the determination whether it works or not. So Bacillus and the Clostridium has endospore. So what does the endospore looks like? Endospore is a multi-layer structure. This outside, exporium or explorium, coat, then by cortex. This is matrix of polysaccharides. Then you go here, cell wall, then cell membrane. Multi-layer. So, it's very rigid. But why it will have a protection structure or protection function? Because right in the middle. Calcium diproconic acids. Right there. Plus very small acid soluble DNA protein. Those two combined together will let the endospore forming is a rigid system and resistant to the harsh environments like UV light, like desiccation, like dry environments. If bacteria has endospore, in the harsh environments they forming endospore, we call it sporation. The endospore, when they form, they could be at a relatively, we call it a dormant status, like a sleeping status. It could be there for a week, a month, couple months, a year, a couple of years, or even 50 years, or 100 years. But sometimes when the condition changes, the endospore will be recreated to become a vegetative cell, we call it a germination. So it really depends. So I want to share with you a story what happened is. This is a true story. I used to work at Kentucky, at Western Kentucky University. I teaching the microbiology there. So one of our friends is a faculty member, Mark Carlson, and he was one day doing some of the dish for the family. Okay. So what he did, in a hot pot, they prepare a hot pot in the morning. In the hot pot, they put water, of course, sliced tomato. Okay, let's say you have a sliced tomato. Then you have sliced carrot. Okay, put some carrots there, like that. Okay, we want some soup, so what we did? The beef slice. Beef slices. Okay, if you have these three ingredients in a hot pot, where are these coming from? Where are tomato and the carrots coming from? They grow in the soil, is that right? Okay, where are these bacteria goes? Of course coming from soil. Bacillus or Clostridium from soil. Lots of the bacteria from soil. And I want to let you know, even you wash them, the wash is called sanitizing. Sanitizing is not going to cure everything. It's usually reduce the bacteria by 99.9%. But whereas that 0.1% could be there. So you need to know that it's not going to cure everything. Because if it's cure everything, we call it a sterilization or we call it disinfection. We'll talk real quick. So you see, he simply did that. When you have a beef slice put in a hot pot, the generated what's a beef soup? It's called a beef extract. And I tell you one thing, one of our major ingredients for bacterial broth is beef extract. Because it has all kinds of protein, all kinds of nutrients. So your here is actually a good environment to cultivation bacteria. Now the hot pot is really bad, is he cooked it. 50 degrees Celsius, around 110 degrees Fahrenheit, for like 6 hours. He go hiking in the morning with his wife and with a dog. That's interesting. This is a typical situation we call low temperature, long time, suicide cooking, if you want to say the terminology. Then what happened? The beef looks good because it's tenderized. This mild heat will kill all the other bacteria. But this mild heat is also a harsh environment for endospore. So the endospore bacteria will grow, will multiplication, and dominate the whole grass. So when the day he comes back, after hiking, so he had a swoop, his wife had a swoop, and the dog had one, and 3 hours later, they all have a vomiting and diarrhea. Then first go to the Bowling Green Regional Hospital. It doesn't work. Then they called his friend at University of Louisville Health Center. And they go there and saved the life. So I just want to tell you, you should never do is low temperature, long time cooking with these plants or fruits or vegetables if they directly coming from the soil. Because 100% they have some residue bacteria, and lots of them has endospore from the soil. This is just 2 examples. Remember what we said. We only know 5% of the bacteria, 95% is unknown. There are lots of the cells or has endospore, we don't even know where they come from. Where are they? Or what are they? And you use low temperature, long time cooking, you provide a perfect environment to killing all the other bacteria, then the endospore forming going to be dominant. Looks like there are no enemies, they just grow. And also the beef extract, the beef soup, give them tremendous amount of the energy and the nutrients to grow. So what is the basic knowledge behind that? When you ever prepare food products, is avoid dangerous temperature zone. What is the dangerous temperature zone? Is 40 to 140 degree Fahrenheit. I don't like degree Fahrenheit. It's pretty much like 4.5 to 60 degrees Celsius, somehow like that. Said very simple, keep cold food cold, keep hot food hotter, much hotter. That's what you always remember. That's very easy to remember. Add one there. 40 to 140 degree Fahrenheit is because of dangerous temperature zone. So that's what's the story for the endospore. And you're always going to remember, endospore is important. It's in our life all the time. It's originally coming from soil. If you prepared using a harsh environments, it may not be support them to grow. It's maybe give them, it may you think they're going to kill, it's actually give them a perfect environment to grow. Okay? So that's what we have here for these sections. These slides are very important is the cell wall structure. So you can put a star there. These 2 slides are important. Okay? The slides right here for the gram-positive cell wall and this one for gram-negative cell wall are more important. And all the others we'll talk later on much more detail. So this is what we have. Okay, so we finish this one. We're going to move on to is our last section. There are lots of we're going to talk. Just to give you a very simple opening remarks, then we will off today. The last lecture slides of the exam 1, we're going to talk about the bacteria grow and also the environments for that. So first of all, very simple, but it's not simple if you talk about the mass. Bacteria cell grow is called binary fission or binary division. What means binary? Is 1 become 2, 2 become 4, 4 become 8, 8 become 16. 2 times the power of n. So then, you should be very familiar with these maths. Sometimes you're going to use it. Okay? We'll have a practice question for you. Okay? That's what will happen. That's called the binary fission. One cell become two cells. Once they grow, from the mother cell, their genetic information is exactly separated and transferred to their daughter cell. And the daughter cell will form in exactly the same structure. Of course, with the exception, there is no mutation. But honestly, it's only going to happen in the lab, in the lifetime, in the real environment. The mutation happens all the time. And as always said, you look like your parents, but you're not the same as your parents. And the same for the bacteria, because mutation will happen all the time. Then we come out with a generation time, the concept, which means how long it takes for bacteria become one cell to two cell, two cell to 4 to 8, 8 to 16. That's called generation time. And we also call it a doubling time. Here tells you something. E. coli is very quickly to grow. Their doubling time, which means the generation time, is very short, 18 to 20 minutes. This means the temperature also, okay? 18 to 20 minutes at 37 degrees Celsius. Not at 4. At 4 degrees Celsius, it's a different story. How about the mycobacterial tuberculosis? Very slow. That's why I said one day maybe one cell become two cells. That is the reason if the patient has presumptive or suspicious tuberculosis symptoms, you can wait for to cultivation the bacteria and to do the determination. That's why we have to use acid fast stain to get the presumptive determination, then use antibiotics immediately. Because if we need 5 cells, how long is it going to take? 6 cells, let's say. How long is it going to take? 3 days could be. Is that right? That could be 3 days, could be 2 days, 2 to 3 days. And nobody can just coughing and sneezing and waiting for 2 days, say, just stay at home. And I'll call you back 72 hours when I get the confirmation of the bacterial culture. So just want to tell you, different species, their doubling time are different. E. coli, Salmonella, Listeria, monocytogenes, or Pseudomonas, the bacteria we use in the lab is very quick growth. But for those human pathogens, tuberculosis almost we can say biosafety level 4. It grows very slow. Okay? Generation time, this related to the mass. We're going to talk about this on Tuesday. Okay, we just stop here, then we talk about cholesterol.